first of all, thank you so much to both the organizers as well as the company for providing me with the opportunity to speak here today. And yeah, thank you all of you for being present here at this early hour and for your attention. So let me get started. I'll start with some historical data. So in 1990, about 90% of drugs failed in clinical trials. Now, we're more than 3 decades later. It seems that that number has changed very little. While progress is being made, of course, in many different fields, it seems that we're still lacking some key issues. Like there are some key issues that are still preventing us from moving up the drugs that actually succeed in clinical trials. And one of the biggest issues that seems to be present here is the lack of preclinical models, like preclinical models that can actually represent human biology in a way that is relevant. And so this makes it very difficult to select the right compounds to advance in clinical studies. And that brings me to the topic of my presentation today, which will cover the preclinical models that we have developed and that can support the drug discovery, selection, and further mechanism of action studies for compounds for your targets of interest. So let me start with a brief introduction of our company, 23rd Biolabs, considering we're actually based on the East Coast, we're not from the West Coast. So we're located on Long Island, near New York, and we were established as antibody discovery and development company, as a CRO, preclinical, and we branched into many other fields of life sciences, including oncology, immunology, many other fields, stem cells, gene therapy, CAR-T, and so forth. And more recently, we also established a new neuroscience division, which I will elaborate on in the next slides of the presentation, and I will also give a brief overview of some of the antibody work that we do. So for the neuroscience platform, we've actually heavily invested in a lot of new equipment. We have live cell imagers, we have electrophysiology, and I'll give you examples using case studies of how we applied these technologies to advance drug discovery studies. And as we wanted to address the unmet need for easy and straightforward assays that are amenable to high throughput screening, that will allow you then to better understand the effect of your molecules or on a particular aspect of the disease, we have developed different types of models relevant, for example, for Alzheimer's, for Parkinson's, for multiple sclerosis, and other diseases. So I understand, of course, that not everybody here is a neuroscientist, so let me just back up a little bit and start with a brief introduction to some key concepts in neuroscience. So first of all, neurodegeneration, which is the key contributor, of course, to things like Alzheimer's, Parkinson's, and so forth, can have many different causes. I will not go into too much detail here. What is listed here surrounding the neuron are many different, things that can happen in the brain. In Alzheimer's, for example, we have a lot of amyloid beta accumulation, protein aggregates. In Parkinson's, it can be alpha-synuclein. There are other factors like Huntington, for example. And then more recently, actually, of course, it's not only neurons that are present in the brain. You have many different cells. You have, besides the neurons, also microglia, astrocytes, oligodendrocytes, and so forth. And all of these can contribute to neuronal survival. These cells can secrete pro-survival factors for neurons. They can be impaired through aging or inflammation. Yes, so they can be impaired through aging or inflammation. So that's also a part of the aging clock, for example, plays a key role there. And all of these factors can lead to loss of specification and neuronal cycle. And of course, we're here to understand which pharmacological interventions can lead to delay or prevention of neuronal death. So as I mentioned, there are many different other cell types in the brain, including microglia, astrocytes, oligodendrocytes. Microglia actually have long been considered the glue of the brain and not to have any other functions, but in the last decades, actually, they have been found to have many other functions, including things like phagocytosis, secreting pro-inflammatory factors, and others. And phagocytosis is basically the cleanup of debris or other dying cells in the brain. And as we see what happens with age and inflammation, we see that phagocytosis actually is decreased in the brain of both older or inflamed brains. And in addition, we also see other phenotypes such as lipid droplet accumulation. And this has been found in many different studies of literature. So when we try to recapitulate these observations that we see in vivo in an in vitro model, we actually find many studies, again, using in vitro models, using a pro-inflammatory stimulus called LPS, that there is an increase in phagocytosis, which is not really, and even in our own data, we see an increase in phagocytosis in a microglia-like cell type, which is not representative what you would expect under physiological conditions, right? You would want to see actually a decrease in phagocytosis. So we set out to investigate what exactly is going on in these cells, and to establish a model that is more representative of what is happening in vivo. And here, what you can see in the images are our lifestyle imaging data. So we have phase contrast here, as well as fluorescence. In blue fluorescence, you can see the cell death happening. Okay, so the videos aren't working, but so we have here the cell death levels indicated in blue, and if treated with a higher dose of LPS, we can see an increase in levels of cell death over time. So we quantify this actually very careful, quantifying every hour what's going on with the cells. We see that after 12 hours, we start to see a significant increase in cell death after this treatment. So that means that any type of assay we do, we don't want to have interference with cell viability. We don't want to have that type of result. So we want to look at the very early time points before 12 hours. So that's what we. There we go. So these are in here. So you can see actually over time that here we do get higher levels of cell death. And then using our phagocytosis assay, and let me briefly explain this assay. We have, again, our phase contrast image here. We will do a time lapse every 3 minutes. We make a picture. And what you will see up here are little dots that will appear here, and these are beads that initially will not fluoresce, but when they are taken up by the cells, they will start to emit red fluorescence, as you can see here. So if you focus on here, you start to see the beads. And then this cell, for example, will start to internalize them, and you will see these are phagocytosing the beads. And what we found is when we change our paradigm, the way we do the experiment, by changing order and concentrations of compounds, we actually find that we are able to actually observe a decrease in phagocytosis. So we are able to recapitulate what is actually being seen in vivo in our in vitro model after really carefully delineating how we need to set up this assay. In addition, we also use this type of system to evaluate lipid droplet accumulation using a different marker called BodyPi. And in green, you can see we have here the lipid droplets in the control. It's very little, but when we treat with LPS, we do see there's a lot more green fluorescence happening. Again, we quantify this at early and late time points, and we use this as a way to screen test compounds. And we actually found, I'm sorry, the font has changed compared to the original presentation, but here we have no LPS and no test compound. Here we have higher dose of LPS. Here we have LPS plus a test compound at 2 different concentrations, and we're capable of seeing a change, or actually a rescue in this lipid droplet accumulation using these assays. Now, these are very simple assays. These are using just a single cell type. It's more or less cell line, which is, again, may not be representative of what's actually happening in the brain. As I mentioned, there are many different cell types in the brain, and therefore we set up a different system which contains different brain cell types, considering, so it contains neurons, astrocytes, and microglia. Here in yellow are the neurons, in green are microglia stained by IGA1, and then here in purple we have GFAP marking astrocytes. And this system can be used, again, to test neuroinflammation. We validated it using, again, the LPS stimulus, which is pro-inflammatory stimulus, and we quantified different pro-inflammatory factors here, such as, for example, TNF-alpha or IL-1A. And in green, you can see the increase in LPS. This is fold change versus control. And we also have here an mTOR inhibitor called rapamycin, and we found there when treating with rapamycin that we're able to attenuate this increased response of pro-inflammatory factors. Of course, this is again only using neurons, astrocytes, and microglia. There is another major important cell type missing here, which is oligodendrocytes. Oligodendrocytes are important, for example, myelination in multiple sclerosis and other diseases. And for that, we actually developed a myelination assay in which we were able to induce myelination in vitro, which is again a very new type of assay. We have here in red the myelin binding protein as control. Push 1 is indicating the neuronal neurites and axons. And then after treating and screening for demyelinating compounds, we are capable of seeing a significant loss in myelination. And on the other hand, we use then this assay to screen for compounds that can potentially rescue this demyelination. And that's exactly what we found here. And this is also quantified in the graphs here, where you can see here is the myelin binding protein, which is the red quantification. And the lower graph is showing the combination of myelin binding protein and the neuronal overlap. So showing it's actually myelination of the neurons that is being affected. And we are able to induce, as I mentioned, significant demyelination and then remyelination. Again, of course, these are primary cells that we use for these assays, and they may not always be easily accessible, or they may not always have the right mutations because a lot of diseases are caused by genetic variants, and therefore this material may be limited. For that reason, oftentimes iPSCs are used, and we have also characterize and set up an iPSC-derived system. We actually use, in the next example, it's an example for Parkinson's, where we use human iPSC-derived Parkinson's neurons, where we validate their electrophysiological profile. So we use a MEA system, which is a multi-well type of format, and every well, at the bottom of each well, there will be electrodes. We plate the cells on top of these electrodes, and then we can measure their electrical activity. And that's also what you can see here in this movie here. So you can see every well is basically indicating the network activity of the neurons. And the more red the signal, the higher the activities. And this gives us more than 40 different parameters in terms of electrophysiological activity. So this is one example of the spike rate, but we also get mean firing rate, synchronicity, burst duration, network activity, things like that. And one of the things that I graphed here is the burst duration, where we saw a significant increase in the Parkinson's neurons compared to the control cells, because we have an isogenic control in split. And what we do here is we use the test compound, which we know is able to decrease this burst duration. And that's also then, when combined with these Parkinson's disease neurons, is capable of decreasing basically the burst duration activity to levels comparable to the control again, which is very interesting. Now, in addition to these monolayer cell systems, we have also developed a 3D system, which is based on so it's organoid platform that we've developed, which we've established this both for midbrain as well as striatal organoids, and we are in the progress of establishing many other brain regions, and not only brain but also other types of organoids, like for example liver, kidney, heart, and others. So what you can see here are the organoids that are developing over time. So they start as initially smaller amyloid bodies. They grow in size and also in the type of markers they express over time. We validated the expression for midbrain of tyrosine hydroxylase, for example. And then here, these are the midbrain and striatal organoids fused together to form kind of like this assembloid, which can also be used to validate compounds or interactions between these different brain regions that are relevant. So, and again, these systems can be used for screening of compounds to validate functions or expression of certain markers. And one more note, so these pictures go up to day 20, but I want to note that we actually kept them in culture for up to 120 days, so they live for a very long time. Yeah, they can be used throughout this time. Of course, the expression of certain markers will change over time. So depending on where exactly you want to, what exactly it is that you want to interrogate, we can select the best time point. In addition to our in vitro platforms, I'd like to mention we also have in vivo platforms available for study of your compounds. We can do ADME, toxicity, and other studies. We have mouse models, rat models, NHP models, and we can also do further customized animal models. And as I mentioned at the start of the presentation, we do not only have a neuroscience platform, we also have, for example, CAR-T, gene therapy, and as really the core of our business was antibodies, I'd like to emphasize that we have a high throughput recombinant antibody production and characterization platform, where we'll go through the project in different steps, which are all very logical. And we have here a very fast cycle. We can go, we have a turnaround time up to within 2 weeks even. We have our proprietary technology with optimized expression strategies, highly efficient and very flexible in scaling. And also the high throughput, I'd like to mention, so we can do many different antibodies in a batch, up to 1,000 plus. And we, here as a case study, I've included one example where we've done, for example, 10 anti-human interleukin 18 recombinant antibodies. As you can see here, this is the raw data for the SDSPH, the seq HPLC, and the SPR data to give you an idea of the quality that we can provide. So what can we do for you? We can help with compound screening, as I mentioned in the neuroscience platform, but again, we have platforms across many different fields of life sciences. We can, for neuroscience specifically, evaluate neuronal viability, but it's not limited to neurons. We can also evaluate the viability of other cell types. Using this live cell imager, this can be very flexibly adapted to other platforms, other cell types, sorry. We can evaluate morphological changes. We also have access to super high and super resolution imaging, so focal as well as super resolution imagers like STORM and STEP. And we can characterize, of course, functional changes. As I've shown you, the neuronal activity is one thing, but we can have readouts in autophagy, metabolism, and other relevant fields. All of these can, of course, be combined to mechanism of action studies to better understand compounds and how exactly they work, and we can perform customized assay development as well. And with that, I would like to thank all of you for your attention. Special thanks to the Creative Biolabs neuroscience team. And of course, if you have any questions, I'm happy to answer them here or at our booth later on. We're at booth number 13. So thank you all.